It's certainly not the same system because this is this microphone here on your computer. Mm-hmm. That's your MacBook computer. So when we turned on this,
Mic check. Hello, hello. Mic check. Check, check. Getting it through there? Perfect. Right. All right. Good morning, everyone. Okay. Um, good morning, everyone. Okay. Good morning, everyone. That's getting better. I'm not going to do it a fourth time. I hope you enjoyed your little break while we helped those who are virtual also be able to enjoy this wonderful presentation. This morning, I am so excited to introduce our keynote speaker. And I'm going to try the French, and she's going to say that I say it correctly, but she's just very nice. Emmanuella Bassey is a senior neuroscience major and French minor, hence my nervousness about pronouncing the French, at the University of Texas at Austin. I had the opportunity to hear her speak last year, and you are definitely in for a treat. There's a lot I can say about her, and I'm going to keep it brief because we really want to spend most of the time with you hearing from her. Through the avenue of LSAMP, we're going to talk a little bit, or you'll hear a little bit about the possibilities of what can happen for you and what has happened for her. So through LSAMP, Emmanuella has conducted neuroscience research in the Brumbach Lab at the University of Texas, and also at, okay, we're going to try, Institut du Cerveau, the Paris Brain Institute in Paris, where she studied the electrical excitability of cancerous brain cells and created various molecular tools to test the functionality of cellular proteins. She is part of the organization Black in Neuro. I know there's some neuroscience majors in the room, which celebrates Black excellence in neuroscience-related fields. Following graduation this May, congratulations, Emmanuella plans to continue exploring the world of research in a variety of ways. I'm just going to tell you about one. She's going to pursue a master's degree at, here's the second one, Université de Toulouse, Troyes, Paul Sabatier. It was better when I practiced, but that's close. Please help me welcome Emmanuella.
Hi everyone, good morning. It's really an honor and a privilege to be here today to get to speak to all of you. Hi, my name is Emmanuella Bassey and I'm currently a senior neuroscience major at the University of Texas at Austin. This morning I have the beautiful privilege to speak to you about what it takes to navigate international research and also promote the view of it from the lens of it being a novel experience that encourages growth, change, and adaptation. So before I dive into my presentation, I just want to give a bit of an overview on what I'll be speaking on today. So first, I'll dive into why even pursue international research, and then I'll segue into what it takes to actually secure an opportunity and go through that process. Then I'll speak about my own experience at the Paris Brain Institute, and then lastly finish off with some goodies that'll include a fun travel video, acknowledgements, questions, and then parting tips as well. So why in the world is it beneficial to pursue international research? International research opens the door to a myriad of opportunities that you could never imagine. For me, it was such a transformative experience because I can stand here today and say, I'm a different person than who I was, I should say, before I departed on May 12th, 2022. International research grants you exposure to the unfamiliar. When you arrive in a new place, a new lab, a new place to live, anywhere, of course you're going to have fear. You're going to be scared. You may not know how to communicate all that you need to in case that country has a different language other than your maternal one. But the beauty of it comes from saying yes to opportunities and being open to what can arise, because you have the opportunity to create core memories that'll last with you forever. Secondly, international research provides both challenge and adventure. When you get to your lab, even if you are familiar with the topic, 10 times out of 10, you're going to come across techniques or different topics that you don't feel deeply familiar with. It may be nerve-wracking and you may have to dig into literature and just follow what your mentors or PI are saying for the time, but at the end of it, you'll be so much more of a different person. You'll be better equipped to face challenges that may greet you in the future. And then once you become more open to that challenge, that's where the adventure part of things kick in, because then you begin to be more curious. You have the seed of curiosity planted in you that can then spur you on towards other opportunities that may arise in your future. Thirdly, international research opens the door to networking opportunities. I'll use myself as an example. During one day at the Brain Institute, a friend of my PI's who was also having a talk that Monday came to speak. Somehow, some way, because my PI has his ways, he got the entire lab to have lunch with him afterwards. I was like, wow, okay, cool. Don't know this person, but let's see what this opportunity can bring about. So we were having lunch. I sat right next to this professor, and I asked him questions about how he was able to find his niche in neuroscience, what inspired him to do a PhD, how he got to where he was, and just thought nothing of the conversation. And then also asked him for guidance for how one is supposed to figure out what they want to do after they finish an undergraduate degree. After speaking with him and then getting advice from him, at the end he was like, hey, and at the time I didn't know what I was going to do after graduation. He was like, hey, if you want to join my lab or so, like feel free to like send me an email in a few months and then we can talk from there. I was like, wait, are you serious? I met you like, what, 10 minutes ago? And you want to exchange with me, be in network with me, and connect with me? That's what doing international research can bring about. Just by being in the room with this person for not even up to an hour, I was able to exchange gain information, and then also have the potential to engage in a new opportunity. Fourthly, international research prepares you for future endeavors. As I said, when you encounter these different research techniques or topics, or even outside of the lab, figuring out how to navigate the train system, go to the grocery store, you are going to be so much more equipped for what comes to your future in case you're thinking about things like medical school, graduate school, entering any kind of job field, because when research occurs in an international context, you become adaptable and more versatile and even more just ready to take on what gets thrown at you. Lastly, and this is the point I want to emphasize the most, international research brings about the opportunity to practice your language acquisition skills. If you're going to a place, knowing at least a few basic words or basic phrases to be able to communicate with people, get around, that can do you a world of good, because that can change how someone interacts with you or how they go on to continue their interaction with you. And the last point on that is that languages serve as bridges, and bridges are how you meet people where they are. Okay, so now that you know about why international research is so great to pursue, how do you go about actually pursuing this opportunity? What does it actually entail? So pursuing international research, I would say, for a few things of advice, when you are trying to apply, make sure that you communicate with someone who can write you a strong letter of recommendation and really attest to your abilities at least a few weeks in advance. This gives them time to kind of just frame you in a holistic way, I would say, that would communicate your strengths and great qualities to whoever comes across your application. Next, begin writing your personal statement or any responses to questions in chunks. If you need to just open a Google Doc and brain dump, do that, because you can end up getting so much more as opposed to just like doing everything at the last minute, which can induce a lot of stress and not produce your best work. Thirdly, get feedback about whatever you write from people you trust. It's always helpful to just hear how what you did from other people is from their perspective, because it could be easy to be like, I think what I wrote sounds great. Maybe this is ready to submit. But no, someone may say, oh, maybe you should like switch out this sentence or use this synonym instead of this to make your communication be better for whoever reads what you write. But on that note as well, I think it's also important to not have too many cooks in the kitchen. Get feedback from at least maybe 2 or 3 people, because you don't want to end up feeling like you have to change everything that you wrote. Fourthly, ensure that any presentation of yourself in a written form or in a digital form is up to date. People want to know what you've done, and they want to be able to find you easily if they can, if you have an online presence on something professional like LinkedIn. But just make sure that the information there is current so they can get the best view of you. And last but certainly not least, be as authentic as possible in whatever you write. You may think that people that are looking at your application may want you to sound a certain way or may want you to communicate yourself in a certain way, but yeah, that may be true to an extent, but above all, they want to know you. They want to know what drives you, what's encouraging you towards this opportunity. Why do you want to do this? How will it benefit you in your future? And as long as you stay true to that, things will be good. Okay, and then a little more on securing the research placement. So once you've gotten that acceptance, what do you start doing? Start finding a lab early. That will serve you so well in the future, because a lot of the guesswork will be taken out of where you're going to be, who you're going to be working with, and what you're going to be studying. Next, make a list of your preferences. I have an example on this on the next slide. But just have a place where everything that you've looked at and everything that you've come across in your loads of Google searching that may happen is documented in a way so that you can go back to refer to it, maybe rank the labs that you find, and help yourself out that way. Thirdly, a lot of people, when you send out emails, are going to want to meet with you virtually. Have questions for them, because that'll show that you're engaged and actually want to become invested, even though your time in the lab may be short, in the project that they potentially bring forth for you. And then lastly, to get a better idea of what it is you're actually going to, sometimes just hearing from the program itself may not be enough to actually quell those fears or answer the questions you may have. So find someone that was maybe in your shoes like a year prior, or someone that can speak to, oh, I've done this before, this is how things were for me, and this is what you can potentially expect in the country or lab you're going to. Okay, and then this is example spreadsheet I was talking about. So this is from circa end of 2021, early 2022. So when I was searching for labs after getting into the LSAMP program, I took it upon myself to just document all the labs that I had found up until the time, and I just organized it by like the university, where it is, what they're studying, the techniques, and for me, techniques were really important because I really wanted to do something different from what I was doing in the lab at my home university. And then also things about like the lab culture, if I got to talk to the PI about it, and things of that nature, but just having things documented. Okay, so now that we've gotten into international research a bit, what in the world did I spend the summer of 2022 doing at the Paris Brain Institute? I had the lovely opportunity of working with B35 neuroblastoma, or cancerous neurons, and seeing whether or not they were electrically excitable. And then following doing that project, I then went into looking at small hairpin RNA-mediated enzymatic knockdown. Okay, so what in the world are B35 neuroblastoma anyway? So as I mentioned previously, these are cancerous neurons that are essentially brought about by the induction of neuroplasms, which is just abnormal tissue that can be able to grow into tumors and then subsequently have the B35 cells extracted from them. Back in the 1970s, a scientist by the name of Dr. Schubert at Yale University injected nitrosoethyllurea into rats, or BD9 rats, and there was induction of neuroplasms. He then isolated the neuroplasms, or the normal tissue, and then got the B35 neuroblastoma cells out along with other types of cells. And then to maintain them for future experiments or investigations, he began to grow them in media, which contains the necessary nutrients and amino acids and vitamins to be able to keep the cells alive to carry out experiments. Okay, so during this first month of my time in lab, my goal was to be able to understand whether or not B35 neuroblastoma are electrically excitable. And this was for the purpose of creating more efficiency in the lab, because the lab that I worked in, the lab of Dr. Jaime de Sanz, it was a synaptic bioenergetics lab, meaning they were focused on how it is that neurons and other adjacent cells use energy or use different kinds of cellular processes to be able to carry out their different functions. But the thing is, when you use neurons for any kind of experiment and introduce genetic material into them, it can take weeks or even up to months to get actual expression of that material and then analyze what it is that the gene you've introduced to that cell is actually doing. So to kind of quicken the process in a sense, I was tasked with seeing whether or not these neuroblastoma were electrically excitable, and then if they were, what would happen would be one, expression of a cellular protein, or a cytoplasmic protein, I should say, known as cytogcamp6F, which would fluoresce and then indicate that the cells are excitable. Okay, so what was I able to see with these B35 neuroblastoma? So I analyzed the B35 neuroblastoma in two different states. In their undifferentiated state, meaning that they did not have extensions known as neurites to make them more like neurons, and in their differentiated state, meaning that they did have neurites that made them more like neurons to be able to see if they were similar to them in excitability. So on this first graph right here, we have on the x-axis the different cells that I used. So we used neurons, of course, as a control to ensure that my experimental setup was actually valuable in testing electrical excitability. I recorded from both the synapses of neurons, which is where two neurons meet and exchange or have communication of different messages via neurotransmitters, as well as the cell body where there can be reception of signal. I also, of course, did the experiment in the B35 neuroblastoma, and what ended up being revealed was that the B35 neuroblastoma, in both their differentiated states and their undifferentiated states, were not as electrically excitable as neurons. And this is also reflected in the graph here on the left. So what I did was grant action potential simulations to the neurons and the B35 neuroblastoma in time, and you can see that the greatest expression of cytogcamp6F was in the neuron synapses, as opposed to in any of the B35 differentiated or undifferentiated cells. Okay, so after that project, in the second month, my PI was like, okay, even though we didn't see that the cells were as electrically excitable as we'd expected, we have another thing for you to do, which was basically investigating the Krebs cycle, which is this well-known cellular process for generating ATP, which is the energy currency of the cell, and seeing what happens when we perturb certain enzymes that result in production of ATP. Okay, so as I mentioned earlier, my PI wanted to know more about this process because it's kind of still a mystery in the field of science as to what neurons are doing with different kinds of molecular processes. How is ATP being shuttled, say, from the cell body all the way to an axon to get release of neurotransmitter. That's not deeply investigated. So my PI allowed me to help get a start on the project in the lab, and what I did was use small hairpin RNA enzymatic knockdown to target different enzymes within the Krebs cycle and see if there was production of ATP affected or not. Okay, so how was our hypothesis tested? So to actually target an enzyme within some kind of cellular process, as I mentioned previously, I used small hairpin RNA enzymatic knockdown, which is essentially a process where you introduce the gene that codes for whatever enzyme that you want to target. And then once you do that, you put it in a plasmid vector, which is basically like the vehicle that allows for transport of that gene into cells you want to introduce it to. And then you amplify the presence of that gene so you have more of it. And following that, you then isolate it and purify it so that you can test it in the cells that you want to. So I was able to get up to step 4, which was following isolation and purification of the Krebs cycle enzyme. I then verified its sequence using Sanger sequencing, and then a future direction being steps 5 and 6 would have been expression, actually, or introduction of the plasmid DNA into both B35 neuroblastoma first for efficiency, and then, if it worked well, the B35 neuroblastoma and the neurons. And then small hairpin RNA degradation would have occurred of a certain enzyme that I targeted, and then we'd see or not changes in ATP levels. Okay, so what could have been done with the work I completed after I left the lab? So in addition to B35 neuroblastoma, there are also other cell lines of B35 neuroblastoma or neuroblastoma in general in existence that have different capabilities. So even though we didn't see a lot of electrical excitability in the B35s, my PI was like, hey, maybe we could look at B50s, B14s, and see if they're more responsive or neuron-like. Another thing that could have occurred, as I mentioned earlier, was expression of the small hairpin RNA DNA within the actual cells, being the B35s or the neurons, in addition to expression of an ATP sensor, which would have the function of reading out how much ATP is retained or not following expression of the small hairpin RNA. All right, and now for the fun part. So this video recaps my time in Europe. I'm not sure where the sound is. Okay, I'll go back so you guys can like see. Oh, okay. They're fixing it. Yeah. Okay, there it is. Yeah. Okay. I can start it one more time. I think I heard it. Yeah. Oh, they're on the computer. Oh, yo, that's okay. Can you guys hear it like slightly? Oh, it's very tiny. That's okay. I can like sing the song if you guys want me to. That works too. But yeah, this is just a video recapping my time. So I stayed in Paris primarily, but on weekends sometimes I'd go out and try and explore. Also sometimes with other people in my cohort, because I think another thing that helped keep all of us grounded was staying in communication with one another. We had a group me and we'd be like, guys, like one person was like, oh, I'm gonna go to like London this week. Like, does anyone want to come? I'm like, yeah, we can split an Airbnb and just like explore and stuff. But it was a really cool experience. So I'll let you guys watch. Ce classe c'est paralysé, quelle angoisse, c'est-tu blanc, évidemment, d'avoir s'éviter dans la glace. Montre-moi l'envers, tu décores à l'endroit, montre-moi la mauvaise, tu sais que cache tout ça. Sorry guys, my singing's not that great. Thank you. Thank you. Cool. So before I leave you guys, I wanted to leave with some parting tips. When applying for any program, such as that is LSAMP, Summer Research Academy Abroad, don't be afraid. Go for it. You never know what can happen. Literally before applying, I was like, Lord, like, I don't know, like, if I'll even get this, but you know what? I'll try. Let's see what happens. Then I got an email, and I called my mom, and I freaked out, and I was like, Mom, like, this, like, actually happened for me. So don't be afraid to put yourself out there and just take on a new opportunity, because it can help you in your future. Next, if you're going to any country, if you're going abroad, try and learn basic phrases, words in the language, because it can help you to get around and also communicate with people. I remember one time, like, I went to go get a beret for like one of my mentors. Like, I walked in and like the person that was checking me out was making conversation with me. He was like, "Oh, like, how do you speak French? Like, where did you learn? Like, what's going on?" I was like, "School." Um, but it was really cool because people are more adept to want to interact with you and exchange with you when you communicate back to them. So it's a really fun experience too, because I'm by no means perfect at speaking at all, but like the effort is what people appreciate. Thirdly, keep in contact with whoever you work with. They could be a very great resource for you in the future when you're applying to any professional program, or just need someone to write you a letter of rec because they already know you. So that's someone in your network. Fourthly, write down everything. Do not keep it in memory, because chances are you're not going to remember what you did. Say someone wants to know, say something like this happens and someone wants to know, what did you do, where you went? If you're like, oh yeah, like I remember studying this mechanism and like I think I went to this place and like did that, people are going to be like, what did you do? They're not going to be able to have a cohesive understanding of what you put your time and effort towards. So write down everything, whether that be in lab or outside of it, however you feel to document the things that you do. And last but not least, be open-minded, be a sponge, be excited. I know like going to a different place, especially if you're going like that with your family, can be really scary for the first time. You're not gonna know how to use the train system. You're gonna get lost. You're gonna be like, oh my gosh, how do I get food? What do I do? How do I use money? But it'll be okay. It's a fun experience, and then by the time you leave, you'll be like, man, I really wish I could go back. But you wanna enjoy every moment of being wherever you are, because such an opportunity may not arise for a while. So. Yeah. But yeah, acknowledgements. Thank you so much again for having me here today. It's really a privilege and honor to be able to just be in your presence. I'd like to thank God for taking care of like just being here, as well as family and friends for pushing me. And then also the lovely Dr. Parker for just being willing to give me a platform to speak to you today. And the amazing, amazing Denise right here, who's been such an instrumental vessel in just getting me to where I am as well. And then my program coordinators, of course, for LSAMP at UT Austin, Timothy Myers and Linda Gonzalez, truly amazing people. And then, of course, yes, the University of Kentucky for having me here, the Paris Brain Institute, the LSAMP Institution, and the National Science Foundation as well. And then the lovely DeLon Sans Lab, truly some of the most amazing people I've ever met. I remember having, literally from the first day I arrived, I was very nervous and I was like, I don't know like how these people will receive me, but we'll just be who we are. Had lunch with them every day, we'd go exploring, they'd give me recommendations on stuff to do, and like I still talk to them. Side note, but one of them had a kid and I got to like buy her baby clothes before she was born, and then she sent me a picture of them wearing them and I was like, oh, full circle. Anyway, very grateful for all of them. And then more acknowledgement. So that's the lab. On my last day, we went to the Pantheon and then went to go get some food together just to commemorate actually getting through the program. And then my lovely cohort, awesome people with Denise in DC back in September. And then Tim and Linda when we went to the LSAM conference in El Paso. Thank you so much again. If you have any questions, feel free to ask them now, or you can email me or also connect with me on LinkedIn. Thank you. Yeah, sure. Oh, sorry.
Does anybody have any questions? And oh, wait, yeah.
Hello, thanks for your talk. I really appreciated it. So my question is, essentially, I need places to look for opportunities like this. Like, surely I don't just Google international research. Like, do you have any recommendations for places to research more specific programs and opportunities?
Like places that also have like similar LSAT programs, or like just where to look for different kinds of international research opportunities, if I'm understanding
The
right?
second one.
For me, I would say I don't really know of anything else other than LSAMP. I know there's also mechanisms through, so UT has like a study abroad program, and like based on like your major, sometimes you can get plugged into different like affiliate programs. But I think like for the most part, for me, it was primarily LSAMP. Because I came across it from actually having a meeting with someone one day, with Linda, and she was like, oh, like there's this like international research like thing you can get to do, and I was like, I've never been out the country before, let's see what this can come of. And then in regards to like places to like go, like if you're like thinking about, okay, what like country should I go to, what lab should I be in? Personally for me, oh yeah, for sure. For me, I really wanted to go to France because thank you, I'd started learning French literally the semester before with no intention of like letting it really go anywhere. Initially, I wanted it to just be a foundation for learning Spanish because I'd taken classes in high school, and then I was like, I don't think I want to do that again. Let's try something else and then see if that can like segue, since French and Spanish are Romance languages. But I think in regards to like finding where you want to go, I think it's very valuable to be somewhere where you know, like even if it'll be like scary for the first few weeks. Of course, like anywhere you go, it might be nerve-wracking. Go somewhere where you feel like after you leave, you'll want to be back there again. Like you'll be thinking about it. You'll be like, oh man, like this was such a great experience. I think I maybe want to do like a master's here, or like do some kind of like program. Oh, something that just came to mind. There are Fulbright programs too. If you want to pursue something like after graduation, that provide funding for I think on the order of a year or two, depending on how much funding you get. And there's different categories like within the Fulbright, so you can do a Fulbright for like research that'll let you like choose your institution to have more autonomy in like where you go and what you do. And there's also one that's set for like I think affiliate programs that are in existence already that accept students that get into the Fulbright program, and you just kind of choose the country you want to go, and there's already a program set up in that respect. In terms of like other things to, yes, if you go to, for example, sometimes the embassies of different countries may have mechanisms on their website, like the Embassy of France has on their website different like kinds of like affiliate programs. Like if you go to a school, like for instance for UT Austin, we have like an exchange program thing with one of their universities that like lets people like go there sometimes, and they also have other like universities that qualify. If you don't end up like doing anything in undergrad, they also have things for like postdoctoral, or for doctoral research and postdoctoral research, based on which you want to go. So I would say looking at embassy websites too is also a pretty good mechanism, for sure.
Okay, and I'm going to put my faculty cap on for a moment and remind all of you that you have campus coordinators. That's your starting point of trying to find some of these resources. But at the University of Kentucky, we invest in undergraduate research. For example, every college has sources of funds that are collaborations between faculty and students. So those are opportunities. Your international offices are waiting for you to walk in. I know that's true on our campus. So what I always tell our students here is go to your campus coordinator. If they don't know, they will figure out who. So that's and I say this to everyone in the room, including campus coordinators, if you don't have the answer, then come to us. Come to me personally, and I will help you find that. So don't let not knowing where to go stop you. I love your points, right? Keep pursuing that, and there are plenty of opportunities. I know most of these campuses well, and your campus coordinators at this point, there are many opportunities for you to be funded to have these experiences. So if this is what you're interested in, start with that campus coordinator, and then the rest of us will help you. I saw other hands. I'll make it back to you.
Good morning. My name is Denise Yates, and I come from the University of Texas System Alliance. And Emmanuella and I have been working together for a little bit. I am one of the co-principal investigators for the LSAMP International Center of Excellence. And our purpose is to help you identify opportunities and resources and things that will help you to identify those programs and those places you can go. And this is a shameless plug for this afternoon, where I will be at a table somewhere, in the Gatton Student Center, and I would be happy to be able to talk with you, give you some ideas in terms of all of those interesting opportunities and some of the resources that are available as well. Emmanuella, I might add, is going to be joining us at the University of Toulouse this summer. She's going to be doing a precursor research experience at Toulouse, and then she's going to be following that with a 2-year master's program in which she's already been accepted at the University of Toulouse.
Good morning, everyone. My name is Alfred. I'm a student at University of Louisville, and the question that I have is, when you were studying abroad, were you able to like publish any work while you were in the labs?
Hi. So no, I didn't publish anything, but there was a paper published while I was in the lab, because before I came, the postdoc and my PI were working together and got the paper out, because my lab was a rather new lab. They started in the winter of 2019, and they explained to me that because of COVID and like other things, it was kind of hard to like get things up. And then also, like, when I went, I kind of didn't have the expectation of like writing a paper or doing anything like too big, because like realistically, okay, I was there for 8 to 10 weeks. My goal was to be able to sharpen different kinds of techniques like I'd get exposed to, but in my home lab, that would be where like my paper writing would be focused on, because I'd been there since like sophomore year. So I would say in a place where you are long-term, like paper writing can be something that you do, but it's also not something that you should be against. Like if you form like a really great rapport with your PI, and like for instance, if your PI is working on something that's analogous to what you're working on in your home lab, and maybe they want to continue the conversation, then I would say, yeah, like continue like doing things like that if the opportunity presents itself. And another thing I would also say on that front, even though I didn't have papers, like if I went to say like Society for Neuroscience conference and I wanted to present like the work I did, I could write an abstract, and then my name would be on it, my PI's name would be on it, and anyone else that contributed to that project, their name would be on it. And that would be something that would go on my CV. But no like full-on papers or anything.
Hi, my name is Enrique. I'm also from U of L, and my question is, I know you had a French minor, and that probably helped you, navigate throughout the city. But I was wondering if you guys also spoke French in the lab, or was it mostly English? Because what if I want to go to another country, but I don't know the language? Like, will that affect the work I do? Because, that's my question.
Yeah, so my lab completely spoke English. So my PI and then 2 postdocs and then a PhD student, they were from Spain, so they would like speak Spanish like on stuff. And then my mentor was from India, and then there were 2 people that were actually like from France. But before I came, my mentor was telling me like we wanted to make sure this environment was like as inclusive as it could be, knowing that like you're not like a fluent French speaker or anything. So they all spoke English to me all the way through. When I was doing like presentation stuff, everything would be in English, but I explained to them that like I also want to try and improve my French speaking skills. So both the 2 like people that were native French speakers, like when we go on lunches or like just explore like the city together, they'd practice French, or I'd practice my French with them and like get corrected and stuff. But in the lab itself, English all the way through.
Hi, my name's Ayana, and I'm also from U of L, and I'm a neuroscience major. My question was, were there any times you failed?
What a great question. Are there any times you failed?
Cool. I felt so much in the beginning and like throughout the process. In my PI, I think that's what I was most grateful for, to have someone that like, even if I did fail or if what I did didn't come to fruition immediately, there was never like, oh my gosh, you can't do this, or like, why can't you do this when like you're supposed to have this background or like know about this thing. There was always a culture of pedagogy, meaning that like if I didn't know something, I could simply ask someone and they'd sit down with me and be like, this is how it works, or recommend papers to me, or send a textbook, or like sit with me and just show me how it worked. Particular instances, and this is very vivid in my memory. So to be able to actually see expression of the protein I talked about, cytogcamp6F, I had to learn how to do something called passaging and then mounting. So passaging was maintaining basically the life of the B35 neuroblastoma because they would double in growth on the dish that I had them in every day. So if I wasn't cognizant of, man, okay, my dishes are like 80% confluent, meaning they're 80% filled, and didn't split them, the cells could die and like compete for resources, and then my experiment wouldn't work. And then on the second part of that, with learning how to use the microscope and mounting, literally God bless them, because they trusted me with so much. The microscope system that they used was a custom one, and they would let me be in there by myself and like learn how to like turn on everything, the laser light, the fluorescent, the stimulator for like the neurons and the cells and everything. But I could not for the life of me mount cells, because what happens is you have this slide that has an opening, and you have your cells on this circular like plastic thing, and you can't see them, but they're there. And you're supposed to like put a ring of like sealant, which was literally just Vaseline, and then another like disc on top of it, and then make sure that like the cells had something called thyroid solution that could keep them alive as you're imaging them, because if not, the cells don't want to be exposed to the air, they can die. I struggled so much with that, you guys. Sometimes I'd break my cells, like this actual slide, because maybe I put too much pressure or force on it. Or like, no, literally, I was like, did I really do that? My mentor would be like, it's okay, just grab another one, because I'd make like multiple in case things like that happened. I remember one day when I was like, okay, I think like, and people would come and check on me too. Like the postdoc would be like, are you doing okay? Like, do you think you got it now? And he was like, don't beat yourself up. Like, you're young, like, you're probably not gonna get this immediately. And I was like, oh, okay. And then, I remember one day I was by myself and I was like, okay, today's the day I'm gonna get mounting right, and I'm gonna simulate these cells, I'm gonna get results, and that would be it. Didn't happen, because went in, did the cells, had like leakage as I turned on like the microscope, and I was like, oh my gosh, I can't see my cells. I'm like, I know I mounted them right. And then ended up like accidentally leaving the electrode, and the electrodes are really, really tiny, in the opening between the stage of the microscope and then like where like I would put other things, and it was stuck, you guys. It was stuck. I was in there by myself, and I was like, God, they're trusting me with this, and like I just probably broke something that cost like $1 bajillion. It was literally, my PI was like, calm down. It's literally like not that expensive, and people break it all the time. But he was like, it's okay, keep trying. Like, you're doing great. Like, I'm glad that like you're not giving up on this and you're continuing to do it. And then before the end of the summer, I was able to do it successfully. Saw my cells fluoresce. And then got to simulate a neuron too, which I didn't include in my PowerPoint, but that was a really transformative experience. So it's really it's not a linear process, I would say. It's very like up and down. But the thing that for me that would happen when I failed, I would like kind of like sit with myself and be like, okay, I know I didn't like get this right, but like what made me not get this right? Was it the way I like put the Vaseline? Was it the way I used the tweezer to like put the disc on top of my cells? Did I have leakage and not like see it before I screwed in the other part of the mount? You know, stuff like that. And then it also helped that like people in my lab were very attuned to like when I'd be like discouraged or like, man, like I really didn't get this. I remember the day after, or the same day when I came out of the microscope room and like was like, oh my gosh, I didn't like get this right. The postdoc was like, do you want to go for ice cream? I was like, what? Now? Like, what do you mean? She was like, yeah, let's go for ice cream. And there was like a cafe like at the on the first floor of the institute. Like we went and like I was about to like pay for it. She was like, no, like I'm treating you to ice cream. I was like, oh, that's so kind. But it got my mind off of like what happened earlier in the day, and I was able to come back the next day, fresh start, and try again. So yeah, it was kind of long-winded, but in a nutshell, multiple times. But just learn from it.
I'm looking at time. Yeah, process matters. You learn a lot. You learn sometimes more from those mistakes than you do from everything going well. And that's, I don't call them mistakes or failures. We call them learning opportunities in our research lab, so.
Hi there, my name is Shirley. I'm the campus coordinator at the University of Louisville LSAMP program. And my question for you is, what do you think were the biggest skills, other than knowing French, that led to your being selected for this research opportunity?
Hi, thank you for your question. I would say this one is pretty basic, but wet lab skills. So before like going into any kind of like neuroscience research or doing what I did at the Brain Institute, freshman like sophomore year, I was in a research lab at school that basically taught like all the basics of research. And it taught me how to pipette, how to do like calculations. That is so crucial because if you miss like how much of a reagent you're supposed to put in or like what the concentration of something will be, that may be why your experiment doesn't work. And you may not know because you're like, well, I'm doing this calculation. Boom, I guess whatever happens, happens. But you can't have that kind of mindset when it comes to science because precision matters. In addition to pipetting, I would say knowing how to read a paper as well was really, really valuable because sometimes like all the information that's in literature is not what you need. You may not need the methods. You may just only need, say, like the discussion and what like a lab thought about something or where like they got their things from as well. So knowing how to read a paper for sure, pipetting. This wasn't something for my lab over the summer, but a lab that I worked for the summer prior, coding is something I wish I would have had like a deeper understanding of because it's becoming more prevalent, I would say, in neuroscience research. Not really sure about other areas, but most likely I would guess as well. And then, what else would I say? Yeah, I think I would say those three: pipetting, knowing how to read a research paper, and then, yeah, coding and stuff. Oh, yes, I forgot about this. Sorry. SRA, so SRA is basically the precursor program to doing SRA abroad. So how that works is you go to any university, so for me it would have been in the University of Texas system, and you do research there over a summer. And that then qualifies you to be able to do SRA abroad. But I think like having that avenue kind of gives you like, it lets you test the waters a little bit because you'll be like in a lab working, and then at the end of the summer, you present at a LSAMP Symposium, get practice with talking to people about your work and what you do and just presenting yourself. And then once you complete that experience, you're then eligible to apply for SRA abroad.
Okay, I'm going to take one final question, but the great news is our guests are here, and I'm sure Ms. Yates or Emmanuella will be happy to answer and engage all day long.
Hi, my name's Sophia. I'm a neuroscience major. I'm also from the University of Louisville. And I was wondering if like there were other opportunities other than research that you can do abroad, like internships or maybe like shadowing.
So you might kind of be able to combine these because I'm listening to questions as I'm walking through. So not just that, but I think that's a great question. But to kind of like think about that typical day that you do too, because while you're getting research experiences, like you showed the video, but there's other things, right? So sometimes you can combine both in that. You can also talk to our guest here today, okay?
Cool, thank you. So I'll start with your question. So I think the thing for me was going to things like study abroad fairs at my university. So sometimes, like, so up at UT, there's this like long hall called Speedway, and sometimes there's representatives from different places that come to talk. And I learned about things like outside of LSAMP, there's one program, I think it's called AIESEC. If you find me after and like want to like get information about it, I can give it to you. But basically, that was a program I found out about that lets you go to like any like region you want, mostly in Europe, I would say. But if you're like on the pre-med track like me and you want to get like shadowing, so what you could do is like through that program do like research, and then you go to like Strasbourg, like which is like in the like northern like France area, and you could like shadow a doctor there. Like the representative was so helpful in just like answering my questions. So there's things like that. But the only caveat with those is like they don't really give you funding. You kind of have to pay for like your time there and fund yourself. So the person that spoke with me was like, yeah, like she worked for like a few months and then like saved up to be able to go. I would also say if your school has some kind of like, so at UT it's called the Office of Distinguished Scholarships and like Postgraduate like help or so. But there's different scholarships, like in addition to the Fulbright I mentioned, like a Rhodes, Cambridge, Marshall, Truman. Particularly for the Cambridge, you can go do a master's or take like a course for a year at the University of Cambridge, and then if you apply like early enough, you get funding for that from them. And then the Rhodes, if I remember correctly, allows you to study English or so, or some kind of like humanities at Oxford for a bit of a time. But yeah, those are the ones like I can remember, but I can like meet with you after as well. And then in regards to like my day-to-day, so yeah, that was a very interesting time. So I guess the way things would work, I had a lot of autonomy as to like when I'd come in for lab, but I tried to be prompt because I really wanted to like show that my PI and like everyone I was working with that I really want to be there and get the most out of this experience. So I'd get to lab by like 9:30, 10, and then work for 2 hours. And then Mondays, there were like seminar talks that we would go to, and then lunch would be 12. And once lunch hits, French people are very adept about like going to make sure they have their lunch. It could be an hour, it could be 2, it could be 3. But during that time, like I would go with the other girls in my lab, and they would be like, "Yeah, you want to go to like the little like cafe nearby?" And I was like, "Yeah, let's try it, even though I don't drink coffee, sure." We'd do like little exploration things, and then we'd come back like early afternoon, and then kind of like grind and like get things done until like 5 or 6. And the thing that put like a time cap on things for me, so I did not have access to the lab past 7 p.m. So if I had to finish something, I had to make sure, I had to really like plan in my head, okay, today I'm gonna do transfection and then maybe just like read a paper because of how long this may take, because I don't wanna get locked out. There was one day where I was doing transformation and then, bless her heart, my mentor had to come back and like let me to the elevator, or let me back to the floor, because the elevator wouldn't like work with my ID, so things like that. And then after getting home, sometimes I'd be really tired, or sometimes I'd be like, you know what, we're literally in Paris, let's go explore. So I would Google things like cool like cafes to go to. In like July, there was like a carnival, and there was also like the holiday on July 14th called Fête de la Bastille, where people like basically like celebrates the storming of the Bastille. And there's like airplanes like that have the French colors come from like their engines, and then there's like the Eiffel Tower, so cool. We'll like change different colors and like shine and stuff. So I would just look for different things to explore. And it also helped where I was living too, because there were students from Columbia that were doing a program similar to mine. So sometimes I'd like meet with them, or like we'd go for lunch. They'd like invite me to like the events that their school had for them. So just, yeah, trying to find things to like explore and just really soak up being in that space before the time came to leave.
All right. So, yes. So I told you she'd be awesome, and I'm going to try to remember everything. So there are a couple of things that I want to reiterate before I give her this UK swag bag to a UT person. One, Ms. Yates will be in the academic fair, so you can ask her questions. 2, Emmanuella will be around. She will also be in the concurrent sessions for a graduate study, but we can be a little flexible about that, so we can have some opportunity to still talk about these international research experiences in those sessions. So feel free to come to those. All right, so, merci beaucoup. C'est formidable. Here's your swag bag. It is full of UK stuff, and yeah, wear that blue proudly at UT. Thank you so much for coming, and we're going to take a short break, and then we're going to go into the poster sessions and the next items on your programs. Thanks so much. Enjoy a short, we might make it a little bit, we're going to make it, shave off 5 minutes and make it a 10-minute break, and then we're going to continue at 10:15. Thanks.
It was wonderful. Thank you. I'm so glad you were so good. I knew you